Translation is the process of protein production. Translation is when a ribosome actually makes the protein using messenger RNA as its recipe. Translation follows transcription. So transcription is when DNA gets copied into a strand of messenger RNA. That messenger RNA will physically leave the nucleus. It will go over to a ribosome in the cytoplasm and attach to the ribosome, and this is where translation or protein production takes place. So before we look at that process, let's take a look at the structure of messenger RNA. Here they're showing us a strand of DNA unwound to show us the base pairs on the DNA molecule, and this DNA was transcribed into a strand of messenger RNA. And so we can see messenger RNA is also just a linear chain of nucleotides that are complementary to the DNA, and there are 4 possible nucleotides that exist in messenger RNA. There's A, U, G, and C. And with that information, the ribosome is going to have to produce a protein. And so the question is, how does messenger RNA encode what amino acid to put where? So if we understand that proteins are made out of amino acids, what messenger RNA has to do is it has to give the ribosome instructions on what amino acid to put in what place using 4 nucleotides. So it can't be a question of just a single nucleotide standing for a single amino acid, because there are 20 common amino acids, so we wouldn't have enough using single nucleotides. Even if we said, okay, well, what if the ribosome was reading 2 nucleotides at a time? All the different combinations of 2 with 4 different building blocks only gives you 16 different possible combinations, so that's still not enough to code for the 20 amino acids. And so experiments were done looking at these things, and so it was thought, okay, logically, what if, the ribosome is reading 3 nucleotides at a time on the messenger RNA, and it was found that sequence of 3 nucleotides on the messenger RNA strand is what's coding for a single amino acid. Knowing that there are 4 different nucleotides that could be put in each of these 3 positions, that gives you 64 different possible combinations of 3, known as a codon, to encode for the various amino acids. So in this picture, they're showing us the messenger RNA strand with brackets, showing us that the messenger RNA is made up of codons. A codon is a 3-base sequence that's encoding that an amino acid, what amino acid should go in that place. The codons, of course, are generated by the DNA, and that same 3-base sequence in the DNA is known as a triplet. So codons are a feature of messenger RNA. If you're talking about a codon, then you're talking about messenger RNA. The DNA that codes for the codons contains 3-base sequences that we call triplets. So like we were saying, there are 64 different possible codons, and this is what's known as the genetic code. This is a chart from the book showing us the genetic code, and you can use it to look up a particular amino acid. And so if I knew, for instance, a codon had the sequence AUG, do you see how the first base is A, second base is U, third base is G, and they all intersect here. So there's my codon AUG, and it's showing me that methionine is the amino acid that's coded there. So because there are 64 different codons and 20 different amino acids, I can see that there's redundancy in the code. And so there are many codons that will code for leucine and so on, which is an amino acid, but there aren't duplicate codes. And so each of these 3-base sequences, the 64 different sequences, are unique. So in translation, once the messenger RNA is in the cytoplasm, a ribosome attaches to it. And one of the first things that has to happen is the ribosome needs to determine how to uncode the messenger RNA. In other words, what the reading frame is. And the reading frame is just the way to correctly interpret the codons on that strand of messenger RNA. So let me just give you an example of what I'm talking about. I put 3 nonsense examples on the screen, and each one of these examples is just using letters as nucleotides. And let's say that this was a strand of messenger RNA. Do you see how if I skipped the C and I started reading at the A, and I read those 3 letters, and I read those 3 letters, that would be different. So that's one reading frame compared to, let's say, if I skipped over the CA and then I started reading 3 at a time after that. And that's a different reading frame than if I just started at the beginning and read the first 3, second 3, third 3. And so what I did, you can see I made, a sentence, cat ate rat. So in this third reading frame, this makes sense to us. We have meaning for those 3-letter words. And this is what the ribosome has to do with the codons. It has to determine what is the proper reading frame, because cat ate rat is very different from, what this says, and it's very different from what this says. And so when the ribosome starts to look at the messenger RNA, what it will do is it will look for this exact sequence, AUG. AUG is known as the start codon, and this will put the ribosome in the proper reading frame. So the ribosome knows that the recipe starts here. This is amino acid number 1. This is coding amino acid number 2. This is coding amino acid number 3. And the AUG, in most books, they'll have it right at the start of our strand, right, because space is short, but there would be nucleotides before and after this point. And so the ribosome has to determine, where should I start reading the message. The way that this process works is that once the ribosome is attached to the messenger RNA, another molecule called transfer RNA, which is abundant in the cytoplasm, will transfer the amino acid to the codon. And so the ribosome technically just holds on to the codon and waits for the complementary transfer RNA to bring the correct amino acid. So each transfer RNA molecule is just a strand of single-stranded RNA that takes on this kind of cloverleaf configuration. It has a 3-base sequence called an anticodon, and that's what's going to try to base pair with the codon. So you can imagine this is kind of like putting a puzzle together. We might try to put a piece in place that looks like it would fit, but it doesn't fit. And if this anticodon doesn't base pair with the codon, no problem, it just floats away and it waits for the next one. On the other end of the transfer RNA is an amino acid. So if the anticodon is complementary to the codon, what's going to happen is this amino acid is going to be donated to the protein being formed at the ribosome. So let's see if we can put this together. Okay, this is our messenger RNA strand. The orange blob is the ribosome, and you might notice that the ribosome is holding 3 codons at a time. So I can see here's a 3-base sequence, there's another 3-base sequence, and here's a 3-base sequence. But the ribosome is really just holding onto those codons, waiting for the transfer RNA that's present in the cytoplasm for the anticodon of that transfer RNA to base pair with the codon. So I can see in this picture that the codon in the middle has a match, and that amino acid is going to get donated to, this is the forming protein, just the linear chain of amino acids. What will happen next is the ribosome is going to move down 1 codon. So we can see that this transfer RNA molecule, we're going to imagine that that anticodon is going to base pair with this codon, which means that the amino acid that it's carrying is going to be right next to this amino acid. And what's going to happen is spontaneously a peptide bond will form between those 2 amino acids. So prior to the ribosome moving down 1, you're adding an amino acid onto the chain. Looking at the next picture, you can see that that has happened, that the amino acid that was in this place has now been transferred, a peptide bond formed, and the whole polypeptide chain is now being held by the transfer RNA in this position. And so what will happen is the ribosome is going to move down one codon, and when it moves down one codon, this transfer RNA that has donated its amino acid is going to be released, and we're still going to hold on to this tRNA with the growing protein. In this picture, we can see that has happened. So this transfer RNA gave away the amino acid. The amino acid is missing. We would call this an uncharged transfer RNA. Here's the transfer RNA that is holding on to the polypeptide chain, the forming polypeptide chain. And here is where we're waiting for a new transfer RNA to base pair with that codon. And what they're showing us in this picture, making it short and sweet, they're saying that this codon doesn't have a match. And so there are 3 codons that don't have a match. They're called stop codons. And when one of those comes into place, what will happen is the ribosome, you can see, will let go of the messenger RNA. Messenger RNA would still be free in the cytoplasm to be translated again into another protein by another ribosome, and the protein. There's the finished protein, which could then fold into secondary and tertiary structure.